Welcome to EPG Pathshala. Myself Dr. Pooja Arora, Assistant Professor, Hansraj College, University of Delhi. I will be talking on a module T cell activation and differentiation. Learning outcome includes introduction, key events in T cell activation which include antigen recognition and downstream events after antigen recognition, co-stimulation which is requirement for T cell activation, T cell differentiation, mechanism of T cell action, CD4 positive T cell and CD8 positive cells, followed by summary. This module helps us to understand how T cells, which are central player in adaptive immunity, recognize and get activated in response to process antigen presented by MSC molecule on antigen presenting cell. How initial binding of antigen to the T cell receptor leads to downstream events such as clonal expansion and differentiation of T cells. What are the essential requirements for T cell activation? And how activated T cell differentiates into effector and memory cell? What are different types of effector T cells and their function? We all know adaptive immune response has important attributes such as specificity and memory response. The T cells and B cell are the major army of adaptive immunity. The T and B cell recognize specific antigen and get activated, in which they undergo clonal expansion and proliferation to form clones of cell which differentiate into effector and memory cell, where effector cell specifically help to neutralize the antigen, while memory cells help immune system to memorize the particular antigen and help to respond at the faster rate in subsequent exposure with antigen. In the present module, we will try to understand how T cell specifically responds to initial binding of antigen, how it differentiates into different types and performs its specific function. First, key events in T cell activation. Antigen recognition by T cell. The single positive CD4 positive and CD8 positive T cell develop in thymus and enter the circulation as resting cell. These cells are present in G0 stage of the cell cycle. The cells which have not encountered antigen are known as naive T cell. The naive T cell have small cytoplasm and little transcriptional activity. The antigen recognition by T cell is majorly done by T cell receptor complex along with the help of additional molecule such as co-receptor CD4 in case of T helper cell or CD8 in case of cytotoxic T cell and addition molecule. T cell receptor complex. T cell receptor is present on the surface of T cell. It has highly variable alpha and beta heterodimeric chain which is chiefly responsible for recognition of processed peptide present on MSC molecule by antigen presenting cell. But TCR is not complete with alpha beta chain. It requires the presence of other chain to express on the surface of T cell. The other invariant chains include CD3 gamma, CD3 delta and CD3 epsilon chain. Together they form CD3 complex and two disulfide link zeta chains. The zeta chain have very short extracellular domain and long intracellular domain. The heterodimeric alpha beta chain along with invariant chain CD3 complex and zeta chain constitute T cell receptor complex. The intracellular domain of CD3 complex and zeta chains have immunoreceptor tyrosine base activation motif known as items. Each item has two tyrosine residue which are the sites for phosphorylation by protein kinase. The two tyrosine residue in each items are separated by 9 to 12 amino acid and presented as this YXXX69YXX, where Y is a tyrosine. As we can see, two Y are separated by 9 to 12 amino acid. Here L represent leucine, I represent isoleucine, X represent any amino acid. Once phosphorylated, the two tyrosine residues help in recruitment of signaling protein that contain two tandem HS2 domain. Alpha beta chain recognize specific antigen, while invariant chain help in downstream signaling events. The alpha beta chains of TCR complex is helped by co-receptor CD4 or CD8 in performing their function. The co-receptor CD4 or CD8 helps in stabilization of interaction between alpha beta chain and processed peptide on MSC molecule while they are binding to invariant sites on MSC molecule. Further, they also help in signal transduction after antigen binding as they are associated with known receptor tyrosine kinase of Src family. Beside co-receptor, there are additional accessory molecules on T cell which interact with additional molecule on antigen presenting cell which further stimulate antigen recognition and downstream signaling events. Together, they all play important role in T cell activation. After antigen recognition, T cell recognize process specific antigen presented by MSC molecule on the surface of antigen presenting cell via alpha beta chain of TCR complex. This binding event also involved CD4 or CD8 co-receptors on T cell as they bind to the invariant regions of the MSC molecule. During this recognition process, the following change happen. Known receptor protein tyrosine kinase belong to Src family such as LCK and FIN present in the cytoplasm or associated with cytoplasmic tail of co-receptor CD4 or CD8 gets stimulated. Also, CD45, which is a common leukocyte antigen, which is also a protein phosphatase, helps in dephosphorylation of C-terminal tyrosine residue of PTK such as LCK and FY and FIN and make them active. The recognition event also lead to conformational change in cytoplasmic tail of CD3 complex chain and zeta chain, which in turn expose its items, that is 2 tyrosine flanking a series of amino acid with particular spacing. The initial binding events lead to activation of downstream signaling events. The protein tyrosine kinase phosphorylate tyrosine site on each items present on cytoplasmic region in CD3 complex and zeta chain of TCR complex. The 2 phosphorylated tyrosine site on each items serve as docking site for signaling protein with 2 HS2 domain such as protein tyrosine kinase phosphoprotein which is JAP70, also known as zeta associated protein of 70 KDA. The JAP70 gets phosphorylated and activated by LCK. The JAP70 then catalyze the phosphorylation of number of membrane associated and cytoplasmic adapter protein which lead to recruitment of several intracellular signal transduction pathway which leads to T cell activation. The important target of JAP70 are the transmembrane adapter protein, LAT linker of activated T cell with long cytoplasmic tail and cytosolic adapter protein HS2 domain containing leukocyte phosphoprotein of 76 KDA, also known as SLP76. The LAT and SLP76 can also be linked via adapter protein GAT. These molecules are responsible for activation of multiple signaling pathway. The importance of these molecules can be understood by the complete absence of T cell activation signaling events in mice double deficient for LAT and SLP76. On TCR engagement and ZAP70 activation, LAT gets phosphorylated on its 9 tyrosine residues. The phosphorylated tyrosine on LAT bind to various effector molecule such as C-terminal domain of phospholipase C gamma 1, P85 subunit of phosphoinositide 3 kinase and adapter growth factor receptor bound protein 2 GRAP2, GRAP2 related adapter downstream of SHE gets. SLP76 also gets phosphorylated and interact with large number of effector molecule such as ITK, which is IL-2 induced tyrosine kinase, and motif that bind to GATs and phospholipase gamma 1. The LAT and SLP76 along with the effector molecule serve to stabilize paroxysmal multimolecular signaling complex that help to mediate the downstream signal transduction. Downstream signaling events, one of the signaling protein phospholipase gamma C carries the signal forward. Phospholipase C gamma gets activated via ITK, a member of TAC family of cytoplasmic protein kinase. ITK is recruited and activated via several ways. ITK is recruited to the membrane by the interaction with SLP76 via their HS2 and HS3 domain and gets phosphorylated by LAT SLP76 scaffold. ITK is also recruited via their pH domain which interact with PIP3 on the inner face of the membrane, which is generated by the action of phosphoinositide 3 kinase by LAT and additional accessory molecule and gets phosphorylated by LCK. The activated ITK phosphorylate phospholipase C gamma 1 and makes it active. Activated phospholipase C gamma hydrolyze phospholipid component of the membrane phosphoridyl inositol 4,5 bisphosphate to generate the secondary messenger such as inositol 1,4,5 triphosphate and diacetyl glycerol DAG. The secondary messenger activate several distinct signaling pathway which results in expression of diverse set of genes required for T cell activation. The three important signaling events are DAG mediated signaling pathway. Diacetyl glycerol generated by phospholipase C gamma 1 diffuse in the plasma membrane and activates a variety of protein which further leads to activation of intracellular major pathway involving RAS and protein kinase C. DAG activate RAS-GRP, which is a guanine nucleotide exchange factor that activates small protein RAS. RAS is a GTPase protein. It is a small GTP binding protein and it is required for initiation of mitogen associated protein kinase, that is MAP kinase signaling pathways. RAS-GRP promotes exchange of GTP for GTP in RAS and make it active. Once activated, RAS activate the serine threonine kinase RAF1, which is a mitogen associated protein kinase, which in turn activates MAC1, also known as MAP kinase, which further phosphorylated tyrosine and a threonine residue on MAP kinase, also known as ERK1 and ERK2, which is extracellular signal related kinase. ERK kinase result in phosphorylation and activation of transcription factor ELK1, ALK1, which is necessary for expression of FOS gene. FOS protein gets dimerized with another protein Jun to yield activator, which is a heterodimeric protein AP1, which is essential transcription factor for T cell activation. Diacetyl or DAG also activates an isoform of protein kinase C, that is protein kinase theta. Protein kinase C theta contains lipid binding domain specific for DAG. It gets activated in the presence of DAG. Protein kinase theta regulate NF-kappa activation. Generally, in resting cell, NF-kappa B is found in cytoplasm in association with this inhibitor known as inhibitor of kappa B I-kappa B, which prevents moving of NF-kappa B into nucleus. Protein kinase C theta causes the release of inhibitory molecule allowing NF-kappa B to translocate into the nucleus, where it promotes the expression of genes involved in T cell activation and survival. Next important signaling pathway is IP3 mediated signaling pathways. The IP3 generated by activation of phospholipase C gamma 1, it diffuses into the cytoplasm and binds to IP3 receptors on the endoplasmic reticulum membrane. It causes a rapid release of a calcium from the endoplasmic reticulum into the cytoplasm and also open the calcium channels in the cell membrane, resulting in influx of calcium in the cytoplasm. The increased cytosolic calcium result in activation of a family of transcription factor called an NFAT, nuclear factor of activated T cell. In resting cell, NFAT remains in the phosphorylated form in the cytosol. The cytosolic calcium binds to calmodulin and causes the activation of calcineurin, which is a protein phosphatase, which dephosphorylate member of nuclear factor of activated T cell family, which translocate into the nucleus. In the nucleus, NFAT can form complexes with other transcription factor, resulting in differential gene expression pattern for T cell activation and formation of different T cell subset. The importance of NFAT signaling pathway in T cell activation can be illustrated by the effect of immunosuppressant cyclosporin A, which inhibit calcineurin, which in turn inhibits NFAT activation, which in turn prevents T cell activation. The importance of DAG and IP3, which are secondary messenger in T cell activation, can be understood by the observation that T cell activation events can be mimicked by Froebel myristate acetate, which is an analog of DAG, iosine, which are the drug which is a mimic of IP3, without involvement of TCR complex interaction with peptide MSC complex. Additional requirement for T cell activation is a co-stimulation. The T cell activation is initiated by engagement of TCR with process antigen on MSC molecule on antigen presenting cell, which is properly known as signal 1. In 1987, Hillenquill and Ron Sequard found that TCR peptide MSC interaction is not sufficient for full T cell activation. The other stimulatory signal beside TCR MSC interaction signal is required for complete T cell activation. The co-stimulatory signal is known as signal 2. The other co-stimulatory signal is provided by the interaction between accessory molecule such as CD28, ICOS etc. on the T cell and co-stimulatory ligands molecule such as B7 family members CD8 T and CD86 on the antigen presenting cell. The one of the most characterized co-stimulatory interaction is between CD28 and B7 family member who together deliver positive signal to the cell which promotes T cell proliferation, cytokine production, cell survival and cell metabolism. After engagement of CD28 by B7 molecule, the tyrosine residue on the cytoplasmic tail of CD28 get phosphorylated by LCK. The activated tyrosine residue on CD28 activate phosphoinositide 3 kinase PI3K, which generate PIP3, which has two important effects. First, it helps in recruitment of ITK via its pH domain to the membrane, where it gets phosphorylated by LCK and LAT SLP76 scaffold. Activated ITK can then phosphorylate and activate phospholipase C gamma 1. Activated phospholipase C gamma 1 helps to activate distinct signaling pathway via secondary messengers DAG and IP3 as discussed before. It helps in the recruitment of serine threonine kinase AKT to the membrane via AKT pH domain. AKT gets phosphorylated and can activate a variety of downstream protein. AKT signaling helps to promote cell survival by inhibiting the cell death pathway and promotes cell metabolism by increasing the utilization of glucose. Thus, signal 1, 2 act in a qualitative as well as quantitative manner to fully activate T cell. Without co-stimulation, T cell becomes non-responsiveness. If T cell receive a signal 1 in the absence of co-stimulatory signal, then T cell comes in a state of a non-responsiveness called energy. In experiment, when T cells are incubated with fixed antigen presenting cell which do not express B7 family members, there is a minimal proliferation of T cell resulting in energetic T cell. Energy can also be induced in T cell when incubated with normal APC which express process peptide on MSC molecule as well as B7 in the presence of Fab portion of anti-CD28 which blocks the interaction of CD28 with B7. Once energetic, T cell become unresponsiveness to normal APC2 which can deliver both the signals required for T cell activation. Therefore, interaction of T cell with antigenic peptide MSC complex and engagement of co-stimulatory molecule with their ligand on antigen presenting cell is mandatory for T cell activation. T cell activation is also influenced by the activity of soluble factors also known as cytokine secreted by T cell and antigen presenting cell. The assisting help provided by the cytokines sometimes referred to as signal 3 for T cell activation. The cytokines bind to cytokine receptor and stimulate cascade of downstream events that enhance T cell proliferation and survival. It also directs their differentiation. T cell differentiation. Once T cell gets activated in response to signal 1 and signal 2, naive T cell will enlarge into blast cell and begin undergoing repeated round of cell division. It divides rapidly for 4-5 days, generating a clones of cell that differentiate into effector and memory cell. The naive T cells, effector T cells and memory cells vary in their phenotype and action as they display broad differences in the expression of surface protein. The function of T cell is majorly done by effector cells, which help in clearance of pathogen, while memory cells remain quiescent and long-lived, but they get easily activated as compared to naive cell. Thus, they respond with a faster rate to the subsequent challenge with the same antigen and are responsible for secondary responses. T cell effector cells and their function. The naive CD positive T cell gets activated in response to following signal. First, antigen specific signal received by TCR complex upon recognition of specific peptide present on class 2 MSC molecule on a professional antigen presenting cell. Second, a co-stimulatory signal transmitted by a CD28 and B7 family members, and a cytokine signal mostly secreted by activated antigen presenting cell, neighboring cell at the site of location of activated T cell, which bind to cytokine receptor and result in proliferation and differentiation of antigen activated CD4 positive T cell into distinct CD4 positive T effector cell. The CD4 positive T effector cell predominantly acquire the ability to secrete factor that enhance and modulate the activation and proliferation of other cell. Thus, CD4 positive T effector cell are also known as helper T cell. The CD4 positive T effector helper cell are very heterogeneous. The heterogeneity mainly occurs due to signal 3, that is cytokine signal. The T cell activation is highly influenced by additional factor, that is the cytokine secreted by antigen presenting cell or neighboring cell. Which cytokine is released by APC depend upon the type of APC, whether it is a dendritic cell, B cell or a macrophages. It also depends upon the neighboring cell such as mast cell, natural killer cell. It depends upon the type of infection, site of infection, etc. The different cytokine activate distinct downstream signaling pathway, which in turn influence the expression of different set of genes, resulting in distinct effector helper cell. The effector helper cells are very heterogeneous in phenotype and function. In fact, based on distinct set of factors secreted by T helper cell, there are well characterized distinct functional subgroup such as Th1 and Th2. The cytokines released by one subset inhibits the development and activity of opposite subset. The phenomena is known as cross regulation. For example, interferon gamma released by Th1 subset inhibits the proliferation of Th2 subset. Similarly, IL-4 released by Th2 subset downregulates secretion of IL-12, which is required for Th1 subset differentiation. Additional T helper cell subset are also developed in the presence of specific set of cytokine, which influence different set of gene in activated T cell and help to differentiate in different subset. For instance, Th17 cells are generated when T cells are activated in the presence of IL-6, IL-23 and transforming growth factor beta, while TGF beta along with IL-2 result in formation of induced regulatory cell. Both subsets have opposite function, where Th17 cell produce IL-17A, IL-17 and IL-22, the cytokines associated with chronic inflammatory and autoimmune response such as arthritis, multiple sclerosis, while induced Th regulatory cell secretes IL-10, transforming growth factor beta, which downregulate inflammation via inhibiting activated T cell and antigen presenting cell. Similar to Th1 and Th2 subset, Th regulatory cell and Th17 cells are cross-regulate each other. They also cross-regulate each other. Follicular helper T cells are the new addition to T helper cell subset. TFHS cells are generated from activated T cells in the presence of cytokines IL-6 and IL-21. Like Th2, TFH cells play important role in B cell health and are found in B cell follicular and germinal center. The effector cytokine released by TFH cell is IL-4 and IL-21. Next T cells are cytotoxic T cell or cytotoxic T effector cell. The naive CD8 positive T cell gets activated in response to the following signal: antigen specific signal received by TCR complex upon recognition of specific peptide presented on class 1 MSC molecules on a target cell, a co-stimulatory signal transmitted by CD28 and B7 family members, a cytokine signal, that is IL-2, mostly secreted by activated helper T cell, which bind to IL-2 receptor, which result in proliferation and differentiation of antigen activated CD8 positive T cell into CD8 positive T effector cell and memory cell. The CD8 positive T effector cell predominantly acquire the ability to kill target cell, expressing the process antigen on their MSC molecules. Thus, CD8 positive T effector cell are also known as killer T cell or cytotoxic lymphocyte, CTLS. Their mechanism include: first, they recognize specific peptide MSC complex on target cell via TCR complex and form cell to cell conjugate. This conjugation event is followed by induction of apoptotic death in target cell. After that, CTL effector cell dissociate from the dying target cell and goes on to bind another target cell. There are two major ways by which effector killer cells induce apoptosis in target cell. First, granzyme and perforin mediated cell killing. After conjugate formation, mostly CTLS starts secreting cytoplasmic granule rich in pore forming protein perforin and servile serine protease called granzyme into the space at the junction between two cells. The perforin molecule contacts the target cell membrane and makes holes in the membrane. The granzyme molecules make entry in the cytoplasm via the perforin made holes or via endocytosis process. The granzyme molecule initiate the event that activate apoptotic pathway within the target cell. Target cells begin to exhibit DNA fragmentation. Another way they can kill the target cell is by Fas2 ligand mediated killing. Here they first form conjugate with the target cell and they mediate killing via the expression of Fas ligand, Fas-L on their surface. The Fas ligand belongs to tumor necrosis factor TNF family. The Fas ligand interact with the Fas, also known as CD95, on the surface of target cell, which delivers a death signal to target cell. The interaction of Fas with Fas ligand leads to association of Fas with adapter molecule FADD, Fas associated protein with death domain, which in turn results in activation of caspase cascade, leading to apoptosis of target cell. Just to summarize, T cell activation is referred to the changes that occur in T lymphocyte following antigen recognition. The naive T cell recognize antigen on MSC molecule via T cell receptor complex along with the help of additional molecule such as co-receptor CD4 or CD8 and addition molecule. It is followed by downstream signaling cascade. Various signal transduction pathway involve RAS protein followed by MAP signaling pathway, protein kinase C followed by NF-kappa B pathway, IP3 mediated pathway, etc. Apart from binding of TCR and CD8 CD4 molecule to peptide MSC complex, additional co-stimulatory signal also known as signal 2 is required for T cell activation. Without co-stimulatory interaction, T cell become anergic. Together, presence of antigen along with co-stimulatory interaction leads to cell proliferation, cytokine secretion and differentiation of T cell into effector and memory cell. T cell activation is also influenced by the activity of soluble cytokine secreted by activated APCs, neighboring cells at the site of location, activated T cell. The assisting help provided by cytokine, sometimes referred to as signal 3 for T cell activation. The CD4 positive T effector cell predominantly acquire the ability to secrete factors that enhance and modulate the activation and proliferation of other cell. Thus, CD8 positive T effector cells are also known as helper cells. The effector helper cells are very heterogeneous in phenotype and function. In fact, based on distinct set of factors secreted by T helper cell, there are well characterized distinct functional subgroup such as Th1, Th2, induced T regulatory cell, etc. The CD8 positive T cell gets activated in response to antigen presented by class 1 MSC molecule, co-stimulatory interaction between CD8 positive cells and target cell and IL-2 cytokine secreted by activated CD4 positive T cell. The CD8 positive T effector cell predominantly acquire the ability to kill target cell expressing the process antigen on their MSC molecules. T effector cells are also known as killer T cell or cytotoxic T lymphocyte. The cytotoxic T cell secrete pore forming protein perforin and granzyme or express Fas ligand to activate apoptosis in target cell. Thank you.